So hey everybody, I'm Raleigh Latham. Welcome back to those who are joining us for the Sustainable Design Masterclass. We're all about bringing some of the top regenerative designers in the world, along with scientists, farmers, and entrepreneurs, to give you a picture of some of the best pathways for the future, whether that's improving your land, your business, or your life. So today, Neal Spackman, alas, he can't be here till an hour into the Q&A. He's meeting with some hotshot Saudi investors for Al-Beta, which is a good thing, but I'll be taking his place. Just reminding you guys, if to get the most out of this webinar, turn off your Facebook. I know you guys were on Facebook, so turn it off. Turn off your phones, because there's going to be a lot of information coming your way. And if you want to get the most out of it, you want to be present for this. So, today we are joined by Peter Mccoy of Radical Mycology. Peter has made it his life's work to study the almost a lost art of fungi, of mycology. Because below all our feet, we are surrounded by the kingdom of fungi. It is the thing that unites all soils, all trees, all ecology, and yet we know so little about it. It has the ability to impact every aspect of our lives. It can make the difference between failure and survival in the driest parts of the world and the wettest parts of the world. So I'm stoked to bring on Peter Mccoy, because he's going to talk about how mycology can be used as a tool in our soils and in our ecology. So without further ado, I'm going to turn it over to Peter. Welcome to the webinar, Peter.
Yeah, thanks, Raleigh, for having me. Thanks, everybody around the world and into the future watching this. I am incredibly excited to talk about this topic. I don't think I've ever actually done a talk on soil fungi specifically, and there's so much to say about this, huge black box of the soil, but even the fungal component, which is a significant aspect of it. And mycology in general, as Raleigh said, is so commonly overlooked and just poorly understood generally, especially amongst well, not necessarily especially, but including soil biologists, permaculturists, people in regenerative agriculture. It's just this big missing piece of the puzzle that so much of my work is revolves around exploring, advocating for, and raising awareness around. And this is a big topic I'm especially excited about because it's so fundamental right below our feet. So yeah, let me dive in, and if we can, let's see, share the slides. If you can, I think, switch it over so I can share my screen.
I need to make you the presenter. Okay, so here we go. Peter Mccoy is going to become the presenter. So let me know that's switching over to you. You got it?
Right.
Awesome. Great.
All right, looking good?
Looking all good. Let's start.
Awesome. So sort of as we were just saying, one thing I think it's really important to point out up front is that mycology, the study of fungi, is actually considered a neglected megascience amongst academics, academic mycologists. On one hand, we nowadays know, after really only about 100 years of good study, probably even 50 years really, looking at fungal ecology, that fungi are integral to pretty much every aspect of the natural world, directly in many ways, sometimes indirectly. And yet we know so little, and also so few people are actually studying it academically. And then at the same time, on the street level, you get very few books enabling people to study the topic. So by and large, it's very inaccessible. Most people don't know much about it. We don't learn much about it, and in our schooling and these types of things in the media. So I always point that out up front to point out that generally, just culturally, we have this awareness gap, and it's nobody's fault, but something we need to be aware of and work collaboratively to overcome, because we're missing out on so much. Over the years of human civilization and expansion and all these things, we've probably impacted fungal ecologies in ways we've really yet to measure and understand. We measure the macro flora and fauna, but we never really look at the fungi. Even today in environmental surveys, most agencies don't consider the fungal component of an ecosystem. And that's something that's being advocated for changing by some organizations like the Fungi Foundation in Chile. But at the same time, generally speaking, even in our environmental advocacy organizations, this is a missing component of the puzzle and analysis. So all that being said, there's so much that we can do as people interested in these topics and regenerating our habitats to advocate for fungi, whether just through conversation or actually through intentional practice. A lot of our human impacts on the environment have, caused extinctions and suppression of certain organisms, including the fungi, including the soil fungi. Of course, a lot of tillage and using chemical inputs in the soil do a lot to disturb the soil ecology and including the fungal ecology of the soil. And just something to be aware of. Maybe for some of y'all it doesn't need to be said, it's pretty apparent, but for others, just making that point clear. So fungi are integral, and the more you learn that, the more you start to recognize that, the more you sort of start thinking about fungi all the time. Of course, I'm biased, but I've seen that in many, many people, and it's a really infectious topic because it's so engaging and it's kind of like a life study when you really start to get into it. Also, it's really important to recognize, without going too deep into this topic, is that essentially fungi have been the soil builders of the planet essentially since the beginning of the Earth. Long story short, essentially the argument goes, and evolutionary biologists have argued, that the first larger complex organisms on the early Earth, on the supercontinent, which was basically sterile rock when it first formed, after bacteria formed, a couple of millions of years later, lichens formed. And lichens today are the first things that show up after lava flow or landslide. The fungal component of lichens are 95% fungal tissue. That fungal component releases really strong acids that can release minerals from the rock, solubilize them, and also, of course, as they decompose, the lichens themselves decompose over time, they become sort of organic matter. And really, the first soils of the whole Earth were built by lichens and primarily the fungal component of that. So they built the soils of the world since prehistory. They enabled plants to survive, develop roots. They built the soils back then, and they still do so much today in this regard. So what are fungi? Fungi, the fungal kingdom, or the Mycetae, is a group of eukaryotic organisms. So they have nuclei in their cells, so they're separate from bacteria, and they are distinguished from animals and plants in several ways. On one hand, they have a unique component in their cell wall called ergosterol. It's sort of similar to our cholesterol, but it's just unique to fungi. Their cell wall is not made out of cellulose like it is in plants, but primarily made out of chitin, and about 10 to 20% chitin and 80 to 90% high weight sugars. Some of those sugars can be medicinal. So their cell wall components are unique. They store their energy as a form of sugar known as trehalose. So these are interesting side notes, but more importantly, more interesting for us is the way that they consume. They do not have a stomach, and they do not photosynthesize. Fungi, sort of like bacteria, get their nutrition through external digestion. They release all kinds of acids and enzymes that are uniquely tailored to their environment. They constantly respond to their environment, and based on what is in their way, essentially, they will adjust the digestive enzymes that they need to eat those things, sort of simply put, break down that food externally, and then consume the byproducts of it through their tissue, through absorption. Within the fungal kingdom, we can see higher up the top of this tree, there's actually several subcategories in the fungal kingdom. Just like there's different types of animals, there's many different types of fungi. But we're going to focus mostly on the three types I'm representing here. On the left, we have a mold, a Trichoderma mold, as a representative of a group known as the Ascomycota. This is our largest group of fungi that we've named the most of. It includes many molds, but also some mushrooms and yeast as well. You can see them sort of hiding on the far left there. Yeast only make up about 1% of the whole fungal kingdom, so they're a very small portion. The vast majority form networks of tissue. They're more macroscopic or larger structured, rather. On the right, we see a large spore, and that's representing a group known as the Glomeromycota. These are soil-dwelling fungi that don't form mushrooms, but as we'll see in a little while, are very, very important for soil ecology. They're arguably the most ecologically important of all fungi, and we'll get deep into them. And then in the middle, of course, are beautiful mushrooms that we're most familiar with in the fungal kingdom. And this one's representing the Basidiomycota. This is the category that contains the vast majority of our more commonly cultivated, wild harvested, edible, medicinal, remediative mushrooms, macrofungi. So we'll talk most about these three categories. The other groups are more microfungi. Not as much to say about them ecologically. Of course, they're important, but just for time's sake, we'll sort of bypass them for today. Also to note is that the fungal kingdom is incredibly large, whereas there's about 300,000 plants in the world, and we've named about 95% of them. Estimates of the fungal kingdom go anywhere from 1.5 to 6 million species, and we've named only about 75,000, or 1.5 to 5%, depending on what estimate you're looking at. So in essence, we know so little about fungi. We've named so few of them, and that's just, of course, naming them, let alone understanding their ecological function and how to engage with them as humans. How do we cultivate them? What medicines do they produce? And how can we apply them into our designs intentionally? So this is where we're at, sort of long story short, mycology being a young, neglected science in this way. It's only about 200 years old. And again, we barely scratched the surface, really largely in this ecological perspective in the last 100 years or less, as we start to fan out in our perceptions. So of the fungal kingdom, as I said, only about 1% form are developed as yeast, and yeast are important in the soils. They likely contribute to nutrient cycling, and they're actually probably a good food source for a lot of microbes and insects. But soil yeast are pretty poorly understood. We can say that about a lot of things about mycology. So we know so little about some of these aspects, unfortunately, and yeast just being one part of that in the soil. The vast majority of fungi, what we're mostly going to focus on, are those that form networks of tissue that's known as mycelium. Mycelium is interesting biologically and physiologically in how it grows, but in essence, it's a network of one-cell thick threads, filaments of tissue that grow from their tip, constantly responding and engaging with the environment at that tip. That's where all the digestive enzymes and acids are released, and the byproducts of the digestion are consumed. And that's really what the fungus is. Most fungi don't form mushrooms. They live as a network of mycelium, like a mold or what have you, fanning through the soil, fanning through the wood, doing all this interesting work that we'll get into. And this is really what the fungus is. Some of them produce different looking spores. We call them different species, but sort of fundamentally, this is the primary fungal tissue. Of course, some mushrooms some fungi rather condense that mycelium into a macro structure we call a mushroom, but even that is just condensed mycelium. It's not different tissue, not a different organ. So here again, it's all mycelium, 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 and that's where all the business happens, sort of simply put. So where do we find fungi? Well, fungi live in all environments. Some of the extremes are represented here, but of course we find them in the more common environments that we inhabit. On the upper left is a photo from a study that came out earlier this year, essentially arguing that of what we know of marine fungi, it seems highly plausible that mycelium permeates the entire ocean floor, which is the world's largest habitat. And just as on land, it's very likely that the soil mycelium, the ocean floor mycelium, which is a sort of a type of soil, if you will, is what takes in the dead ocean life and churns it up and breaks it down and recycles those nutrients and releases it back to the whole ocean. And of course, a lot of those nutrients end up back on land. So fungi are fundamentally at the bottom of much of the world's food web in this way, and they do this nutrient cycling role as one of their primary functions on the terrestrial plane. On the upper right, we see a cryptobiotic crust community in desert ecosystems. We'll talk a little bit more about them later. We can actually try to cultivate these, but they're very important in these dryland environments because they stabilize soils. These are communities, of fungi, lichens, cyanobacteria, microplants, and they basically create micro pits and valleys that can collect water, hold habitat and space and shelter for seeds, enabling them to establish in these environments. And even when we get deeper into it, as we'll see later, they actually fix a lot of nitrogen for these ecosystems, and the fungal component helps translate and translocate that nitrogen and other nutrients throughout those ecosystems. So these really complex communities take a very long time to develop. If you go to these environments, you often see signs saying don't bust the crust, because they take hundreds of years to develop. And very likely much of our, I imagine that much of our desert ecosystems in North America, other parts of the world have these communities, but human intervention and just animal compaction has destroyed a lot of them. So just something to consider in deserts about how we can restore these communities to help stabilize the soil, reduce erosion, and increase water holding capacity. At the bottom, sort of hiding there, is a photo of the ground crust, if you will, I guess the soil of Antarctica. And what we're seeing is just below the surface layer is a black layer of melanized fungi that filter the intense UV of the sunlight in Antarctica for the plant component, the algal layer, right below it. So this is a type of lichen that hides inside of rock. They're known as cryptoendoliths. And my friend studies these, and these are much more abundant than plants in Antarctica. There are about 2 flowering plant species and over 430 lichens or lichen-like associations in Antarctica. Much of them look like this, hiding in the crust layer, sort of the soil, if you will, of Antarctica. So here again, we find fungi in all these extreme environments, incredibly resilient, incredibly abundant, and integrally playing roles in nutrient cycling. Of course, we're more focused today on the terrestrial environment and our terrestrial soils. And probably a lot of you know, but maybe some of you don't, that soils form sort of from the top and from the bottom. Down below, there's the parent material, bedrock, that is slowly over the eons worn down into sort of smaller and smaller mineral bits. And part of that we think is through, of course, erosion, and some microbes can slowly break down minerals. But so do fungi, as we'll see. They very much are involved in releasing minerals from clay and rock, and I think play a big part in the slow dissolve of parent material into the mineral component of soils from below. And then up top, we have the organic material that falls down from plants and everything else, and that slowly degrades into the more organic component. Fungi, as we'll see, are integral to that decomposition. And then in the middle, we get a gradation of the blend of the two. And different types of soils are basically different ratios of these components. So how do fungi contribute to both ends of the spectrum, the mineral end and the organic end, and what do they do once that soil is formed? This is what we're focusing on today. So on the mineral end, fungi, on one hand, both dissolve rock, if you will, break it down, release those minerals, and on the other end, they can actually recombine certain elements and create minerals in the soil through biomineralization, as we call it. So on the left, we're looking at a topic known as geomycology. This is a term coined by a researcher by the name of Gadd. And many types of fungi, many microfungi, molds, have been shown to break down all kinds of rocks. So in the middle, we see many types of rock that fungi have been shown to grow on, release minerals from, primarily through the release of really strong acids that, again, they release from the tips of that mycelial network. Oxalic acid is a common one, malic acid, citric acid, and a few others are quite abundant. And these really strong acids, they have a strong KPA point or pKa point, for the chemists out there. But it's really strong acid, and it can release these minerals quite well into the environment, both for the fungus, but also for the plants it associates with. The 2 molds on the bottom have been shown to release aluminum from alumina silicates clays. So that's pretty interesting in my opinion. So just like some microbes have been known to solubilize and release minerals, and there's a lot of study around bacteria in this regard, fungi do it as well. But as with so many areas of mycology, this is just really poorly looked at. And then that information and what we know just has not translated out and rippled into the ecologists and soil-minded folks amongst us. So even though you haven't heard a lot about it, it doesn't mean it's important. I think that's just, again, I'm hitting this point home hard because so much of my work is saying, yes, this is maybe new information to you, but it might be incredibly important to our environmental analysis and our perspectives. We just need to look more at it. We need to talk about it more and really analyze the depth and magnitude of these kinds of concepts. So there's that. They release a lot of minerals, and at the same time, they help recombine minerals or real elements in the soil and create different sinks. So some strong examples are the combination of calcium and carbon into different compounds like wheatolite, and whewellite is another one. These are 2 really common carbon minerals in the soil, some of the biggest carbon sinks. These are likely produced by fungal biomineralization. So what we're seeing in the top picture with the A is actually a fungal hyphal thread covered in wheatolite crystals. So it's a pretty amazing photo, in my opinion, So it's producing this, creating a sink of calcium and carbon in the soil. Below a paper I just actually came across researching for the webinar, it just came out a couple months ago talking about how it's very likely that fungi actually produce calcite, another important calcium and carbon sink in the soil through mineralization. Bacteria have been thought to do this, and they do it to some degree. But again, the fungal component, this is a new paper saying nobody's really looked at this notion. Do fungi create the calcite in our soil as well? And many other minerals in our environment, very likely fungi produce or contribute to the production of, helping sink these minerals and then later perhaps releasing them as needed. So as my friend says, fungi are nature's greatest chemists. They do so many things chemically that are unparalleled by bacteria, animals, and plants, and this is just one example of that. They also influence soil pH in a variety of ways. One common example is that various types of soil fungi release this oxalic acid, which of course is acidic, so they release more of that, so the arrow is going up. And they can also uptake ammonium, which also contributes to the lowering of pH. And in lower pH environments, many minerals and metals and trace elements are more available to plants. So it helps lower the pH. This is why fungal soils are a bit more acidic. They contribute to that acidification so that plants can get the nutrients they need. Arguably, if you want to think about it in this sort of meta picture, with fungi sort of contributing to the growth of life on the terrestrial plane at least, in many ways, I sort of see it as fungi sort of sculpt our environments. They really contribute to the flow of nutrients, and they fundamentally determine what plants and ultimately animals can live in an environment. And this is in one way where they're really directly contributing to so much of the soil design and then the environmental design that comes from that. And then on the other end, we have the decomposing fungi that break down the organic matter. And this happens in a myriad of ways. Fungi are responsible for 90% of decomposition on the planet. And bacteria primarily fill in the gaps. They really eat the smaller stuff that fungi have released from the more complex compounds. Of course, bacteria can readily eat simple things like sugars when they're available, but stuff that's locked up in, say, lignin, which is nature's most complex compound, only fungi can break down lignin. Lignin is what makes wood hard and rigid, and the fungi make that available. So we have many different types of decomposers. What I'm showing here are, of course, on the upper left, beautiful reishi mushroom. Many of the mushrooms we cultivate commercially for food medicine are decomposers. They don't have symbiotic relationships. They just eat dead things. Some fungi can eat, even attack living organisms like nematodes, as we see in the upper right. Some will actually eat micro insects like springtails and actually attack them living and suck out their life, if you will, and feed that to their tree partners. But primarily when we're thinking about soil building, the decomposers are integral to that because, of course, they're decomposing, but also adding many of the important components to the soil. Specifically, wood decomposers are a major piece of this puzzle, wood being a major nutrient store and trees, of course, being a major component of our land systems. There's two main types of wood decomposers. There's really three, but there's two we mostly focus on. On the left are the brown rotters that break down cellulose and hemicellulose, leaving behind the blocky lignin. This brown rot material, cubical brown rot, CBR as we call it, might actually be a really important component of soils. It's like a sponge, kind of like nature's biochar, as my friend says. Holds a lot of water, nutrients, roots will grow into it, microbes are found inhabiting it, mycelium will grow into it, sort of sucking out the nutrients that it stores. And it's often overlooked in soil surveys. So again, we miss out on this component that might actually be pretty important. On the right, we have the white rot, which is produced by the only fungi that can break down lignin. The only things in the world that can break down lignin are about 2,000 species of fungi. They eat the lignin, leave behind the stringy cellulose, and this has actually been argued to perhaps contribute to the production of some of the humic acids and humic substances in our soils. Of course, we don't quite know how humic substances form, and it's been argued that perhaps some of the byproducts of delignification might be part of that structuring. The byproducts of that look very similar to these humic substances, so that's a theory that's out there. It's kind of interesting. Okay, so what kind of fungi do these things? Well, we have a lot of molds in the soil, and some are better studied than others. One to be aware of for gardeners and things like this are the Trichoderma species. These are a very ubiquitous genus of fungi around the world. Several species have been heavily investigated for their ability to primarily defend against other fungi. These are actually major check and balance in the soil, and even though they don't look so pretty, they're actually very important for fighting off other fungal pathogens. So you can easily grow this mold. It's a mold, and it grows on almost anything sugary and wet, and then you can apply it to the root zones of plants, and it will defend against pathogens. Of course, you have to try to bring that in before the pathogen gets there. The pathogen's already taken over. It's kind of hard for the trichoderma to win, but it's always worth a shot if you're in a kind of do or die scenario with your plants. And they can also be added to compost piles and actually speed up the decomposition of compost piles quite rapidly. They have really strong cellulases. They break down plant matter very quickly. And there's a lot of research been done with trichoderma in both these regards. So easy to look up and learn about trichoderma if this is new to you. Where we're mostly going to focus on for a good little while now are the fungi that are perhaps the most studied in soil systems, which are the mycorrhizal species. Myco meaning fungus, rhiza meaning root. These are fungi that symbiotically form a relationship with plant roots, clamping onto the roots and exponentially increasing their surface area 10 to 1,000-fold. So we can see that in the photos in the upper left, where the uninoculated plant with its roots can't really access a lot of nutrients. Roots in many ways are, in my mind, like porous anchors. They hold the plant in place, but they haven't evolved to do the chemical work needed to transform nutrients and bring them in. They've primarily evolved to rely on fungi and other microbes to do this work for them. The oldest plant fossil with roots has this relationship from about 450 million years ago, and today 90 to 95% of all plants form or can form this relationship, and many of them, like melons, oaks, pines, citrus plants, grapes, will not survive in the wild without these mycorrhizal partners. In our croplands, we have to heavily fertilize and provide all kinds of defense chemicals and stuff to sort of try to mimic what the fungus naturally does. Many of our crops will significantly benefit from the introduction of these fungi because they not only fan out and access nutrients and do all this chemical work that the plant just can't do, they also draw in water, enabling greater drought resistance. They can stabilize the plant in a bunch of ways, as we'll get deeper into. And even beyond that, they connect many plants together in an intact forest system. One mycelial network could connect potentially hundreds or thousands of species of plants together in what we call a common mycelial network, a CMN. And intelligently, so it seems, or at least very efficiently, distribute nutrients amongst those organisms in ways that keeps everything going and everything alive. This is how the baby trees survive in the depths of the forest. It's by the mycorrhizal fungi providing sugars from the mother tree that's reached the canopy. There's other studies that have shown that, say, when one plant gets hit with aphids, the mycorrhizal fungi will translate some sort of information to other plants in the system, and those plants will start to produce anti-aphid compounds even though the aphids haven't gotten there yet. And that's actually a pretty significant influence, kind of rippling out to whole insect populations ecologically, all these dimensions we can't exactly fathom. So they're integral to forest health, and we'll get deeper and deeper into this. There's only about 6,000 fungal species that form this relationship that we know of. Again, 90 to 95% of plants form it. And in a healthy soil system, every gram of soil might have a mile or so of fungal mycelium. So it's kind of like our capillary system, very efficiently packed, very high surface to volume ratio, fills a lot of space and accesses a lot of nutrients and does all this interesting stuff. There's about 7 types of mycorrhizal fungi that we differentiate here in the drawing, representing 5 of the more common ones. And I won't go into the details, but you can kind of see that it's based on the structures they form, how deep they go into the root, and if they penetrate into the root cell wall itself. The formation seems primarily because when the plant is starved for nutrition. If the plant is overly fertilized, it doesn't need the fungus, and so if we're dumping inputs into the soils, then the relationship won't form. So if you buy products that tout having mycorrhizal fungi, but they're also nutrient rich, it's kind of a marketing scheme that isn't necessarily going to benefit you, at least in the mycorrhizal regard. Primarily, it seems to happen when the plant is sort of starved, especially of phosphorus and nitrogen. And under those stressful conditions, the spores need to be introduced to the root zone of the plant. You can't top dress the soil, and the spore will germinate. There'll be a sort of a communication, and the plant allows the fungus in, if you will. There's different types of entries. Some fungi only form on the surface of the root and form a thicker layer, and only go into a couple layers deep into the root, and these are known as Ectomycorrhizal fungi on the far right. They don't necessarily penetrate into the root cell itself. They can maybe enshroud a cell, but they don't go into the cell. And this is most of our wild mushrooms and truffles, and we'll talk more about them. On the left, we have the representative of the Endomycorrhizal fungi. These are fungi that usually go deeper into the root, but more specifically and more importantly, actually penetrate through the cell wall, not the cell membrane. So they don't go into the center of the cell, but they enter the cell to a degree and form different structures to exchange nutrients with the plant partner. And there's several types of these Endomycorrhizal fungi. The most common, and the one you might hear about the most, are the arbuscular mycorrhizae, being represented here, but there's several others we'll talk about. Generally speaking, mycorrhizal fungi do lots of things in the environment. One of their main roles is providing nutrients, whereas on the left, we're showing that our attempts to provide nutrients to plants, at least artificially, not talking about organic fertilizers and natural fertilizers. And the most extreme, we strip mine phosphorus rock in this example, dip it in acid to release the phosphorus, all to create a water-soluble form of phosphorus that the plant can just naturally and passively absorb. Of course, fungi naturally transform this in the soil. They'll use their acids to release phosphorus from rock, provide it to the plant, and we can significantly reduce our fertilizer input and, of course, all the environmental impacts that can come along with those inputs. So the plant provides a lot of nutrients, lots and lots of studies leading to higher nutritional content, medicinal quality, flowering rates in the plant, overall greater growth rates and survival time of the roots, and even shortening the time it takes the plant to reach maturity. They also do a lot to defend the plant, both as a physical barrier when the mycelium is enshrouding the root tip, but also by releasing all kinds of antibiotic compounds. They can release their own. They can influence the plant's own defense mechanisms. Like I said, with the insect herbivory thing, that's the vast ecological effects there on the third point. The fungus itself might just compete for nutrients and outcompete competitors. It can also stimulate the growth of other beneficial microbes, overall increasing the plant health, and there again reducing our need for pesticide inputs. So lots of studies have been done with mycorrhizal fungi, especially the arbuscular mycorrhizae I just mentioned, and we know a lot about them. So these arbuscular endomycorrhizal fungi are usually just called them the AM fungi or AMF. They are endos, meaning they go into the cell wall, as I'm showing the lower right there. That's an actual photograph of an arbuscule, this branch structure that enters the cell wall. But they're not the only endomycorrhizae. So some people just call all endos, or all, it is simply call them the endomycorrhizae, but that's vague. It's not specific because there's actually multiple types of endos, so just clarifying that. More specifically, they're the arbuscular mycorrhizal or arbuscular endomycorrhizae, if you want to be really specific. There's about 169 of these species or morphospecies. They're in the group the Glomeromycota I mentioned at the beginning, and many of them are ubiquitous around the world. That being said, there's many different types of these. In the center, we can see their large spores are actually packed with hundreds or even tens of thousands of unique genetically distinct nuclei, which is fascinating and very strange biologically, but for our purposes translates to the fact that they got a lot of genetic potential and they can utilize that to adapt to their environment, basically shift whatever sort of genetic expression they need based on what their environment demands. So this kind of translates into not importing some of these soil fungi. It's usually better practices in many regards to work with the local fungi because they're going to be adapted to your environment. This translates to all fungi in cultivation, but here in this soil example is a strong, strong one. On the left, we're showing a photo of the first plant with root fossils, or first fossilized plant with roots from about 460 million years ago. And here we find on its roots an arbuscular mycorrhizal fungus that looks very almost identical to species we find today. So it's a very ancient relationship. Hasn't changed necessarily a ton over all these eons, and incredibly efficient, found throughout the world. They're found on every continent except Antarctica, in forests, deserts, aquatic environments, grasslands, salt marshes, pretty much everywhere. Generally more alkaline soils, more mineral rich. They're usually in the more mineral rich layers of the soils, or perhaps contributing to some of that mineral release. And they're the ones that really connect plants together. They're the generalists that don't discriminate, and they're connecting 85 to 95% of all plants. They're super densely packed in 1 square meter of soil. Their surface area might cover 90 square meters. So they're accessing a lot of that soil. Lots have been said about them. They are the most studied plant symbiosis because of their incredible importance. They've been shown to increase many crops' production rates, as we see in the statistics. In some, when the plants are stressed or starved, they can provide up to 80% of the phosphorus input. Their benefit can help us reduce our fertilizer inputs significantly, especially phosphorus and nitrogen. They can even reduce nitrate, which is a pretty metabolically taxing process. They've been shown to be defensive against Fusarium and Phytophthora diseases, very common root rot issues. And they're a very important water source for desert plants. One of the most important things about the arbuscular mycorrhizae is they produce a unique sticky protein on the surface of their mycelium called glomalin. This is like a super glue of the soil, and it's very persistent, lasting upwards of 50 years in the soil, likely due to its iron content. It is unique to these fungi, and it's produced on the surface probably to hydrate and lubricate them, but it also helps stick the soil together and basically creates macro aggregates that enable the greater tilth and structure in the soil that we all appreciate for water and air percolation, root movement, worm wiggling, etc. Really important for that aspect. Also helps reduce erosion, desertification, thereby increasing water holding capacity and suppressing fires. Really integrally contributing to the structure of soil, the health of soil overall. Remediation-wise, it can actually bind up some metals, and it's been argued to, and actually been shown to be one of the major carbon sinks in the soil. It actually might be one of the biggest carbon sinks on its own right, this protein, but when you take into the mycelium itself, which is a huge fungal, huge component of the soil and a huge carbon component, fungi and the glomalin might actually be one of the biggest, if not the biggest, carbon sinks in the soil. So when we're talking about trying to sink carbon out of the atmosphere, we need to think about getting more soil, more fungi going in our soils, not necessarily trees. I mean, trees come along with that, but we never want to forget the fungal component. So humic acids only sink about 8% of the soil carbon for relation. On the bottom, we're showing you can actually extract glomalin. There's processes out there, a little bit technical, and we're showing when you extract it, what does the soil look like? So it actually adds a lot of the color and shading to the soil, which is interesting. We can cultivate them. So on the left, I'm showing a photo from the Rodale Institute. They actually spent 6-7 years working with farmers to figure out ways to grow a lot of AM cultivation, inoculum cheaply and abundantly for farmers. So you can look up the Rodale Mycorrhizal Cultivation Protocol. It's pretty simple, straightforward. Basically grow a grass or allium or some quick-growing plant in a pot or bag, and the roots and mycelium down below, and harvest at the end of the season and apply to your crops the following year. Pretty straightforward. A lot of benefits, and especially if we're working with the local fungi, local soils from a healthy forest, and amplifying that soil and those mycorrhizae up. On the right, I'm showing more technical processes that the West Virginia University is researching. They have a big arbuscular mycorrhizal research department where you can actually sieve your soil and separate out these spores, quantify their numbers and diversity. Good for soil analysis, also good to track the efficacy of any attempts to cultivate these fungi and increase their abundance in depleted soils. It's a nice metric to actually see how the fungal spore loads are increasing, changing over time, etc. So there's more information there. This is all covered in my book, which Raleigh didn't mention, but I have a lot of information about these fungi in my book, Mycology. A couple other types to give a shout out to. The Arakoid Endomycorrhizal fungi are primarily restricted to Arakoid species or plants in Aracaceae. They're mostly Ascomycota, and they mostly associate with the Erica, Calluna, and Vaccinium, so like blueberries and relatives, heather plants. They're more in the acidic soils that these plants prefer, grasslands, tundras, heathlands, boreal forests. In some heathlands, the soil can be at 2.5 pH, like lemon juice, and yet these fungi enable plants to survive. They're doing all the work we've talked about so far, and at the same time, they can actually act as the primary decomposers of that whole ecosystem. So if the same plant they relate with dies and falls over, that fungus digests it and returns its nutrients to its brethren. So these have actually been called the drivers of these whole ecosystems because they intimately and directly control the flow of nutrients. Pretty powerful work. And yet again, these fungi, for all their interesting facets, are pretty poorly understudied. We do know that they can benefit blueberry plants in our own garden when we bring in the right type of Arakoid fungi, not just our generic other fungi, Mycorrhizae. They're not all the same off the store shelf. And there are some products coming out with Arakoid Mycorrhizae specifically, so you can look out for those. A couple other types of fungi, mycorrhizal fungi, just to recognize, all the monotropa plants that don't photosynthesize are fed by mycorrhizal fungi. They form a other type of association known as the monotropoid Endomycorrhiza. Basically, it's the fungus connects to these monotropa plants, the pink, the white, the kind of stranger looking plants in the forest. And then on the other end of that mycelial network connects to a tree or other plants and channels nutrients to the monotropa to feed it. Pretty interesting relationship. It's usually seen as a one-directional relationship, but it does seem that the fungus might get some benefit from it, or at least it can go both directions. Studies have shown this. Interesting fungus that does this is our king bolete or porcini, a delicious and prized gourmet edible, actually forms this relationship. On the right, we have the Orchidatious Endomycorrhizae, which all orchids form when they germinate. All orchids do not photosynthesize. They need these fungi for their survival, sometimes for years, sometimes for the whole life of the orchid. These fungi pack their mycelium into the root cells, feed the orchid, and do similar things. They can either be digesting wood or connecting to other plants to retrieve these nutrients. Many common woodland mushrooms, like Russula species, do this. A really interesting one is the Armillaria or honey mushrooms in the lower right. Often seen as a pathogen, a problem, a parasitic mushroom actually feeds and supports a lot of orchids at the same time that it kills off some trees. So interesting dual relationship there. Our most common, or probably our most familiar mycorrhizal types of fungi are the Ectomycorrhizae that form mushrooms and truffles. These form a thick layer of mycelium around the root tips. These are known as a mantle sheath, so you can see it's really thick. There's no root being exposed. They're mostly in the more organic rich upper layers of soils, usually higher elevation, older forests typically. This includes our chanterelles, our boletes, our gourmet truffles. These are the ones we wish we could cultivate, but as of yet, only a few species we've dialed in, mostly ones that don't seem to be as dependent on appropriate or really specific soil conditions. This is a big hurdle for us growers in the future, in the present as well, is trying to figure out the right soil conditions, microbial, insect communities, other plant communities that, say, chanterelles need to fruit in our own backyard. Right now we can't do that consistently. That's why they're wild harvested. Whoops. There are some species that we can cultivate, Ectomycorrhizae, that are commonly cultivated for silviculture, tree plantations, like where I live, Douglas fir and western hemlocks. They're all inoculated with commonly Lacaria mushrooms, which are actually edible and medicinal, so it is an extra crop. They're just not gourmet. Or say Pithelicus tinctorius on the left there. Not so pretty, but actually a very important mushroom to trees around the world. Very heat tolerant, can survive in 104-degree Fahrenheit soils, actually quite beneficial to trees in those kind of hotter environments. Establishes with many trees around the world, very common species, doesn't always fruit, but it's very common in soils, and it establishes quite readily with trees, so it's an easy one to work with. Other common species we can grow to inoculate our trees are in the Rhizopogon, Hebeloma, and Swillus genera. Pithelicus is also important because it can associate with over 50 trees and tolerates soil pHs anywhere from 2.6 to 8.4. That's a huge range, so it's an incredibly tolerant fungus, well adapted to helping trees. In desert environments, desert truffles can support a lot of plant species, especially Helianthemum shrubs, and these desert truffles are a protein and medicine source and traditionally consumed by Bedouin cultures and many other cultures in North Africa, the Middle East, Mediterranean climates. spoken about for 1,000 years by the Prophet Muhammad, this type of thing. And we can cultivate them. It's not necessarily common practice. I'm certainly interested in seeing more folks, say, in North America taking this on and seeing how viable it is as a industry and supplemental food crop in, say, like, California, Southern California, or Eastern Washington, things like this. I don't think anybody's really looking at it as far as I'm aware. Probably a lot of intriguing stuff possible there. Seems to be pretty interesting as far as I've read. Would love to see other folks exploring this topic. But desert truffles, big history, pretty big like a potato, and supposedly tastes really good. A couple other fungi just to be aware of. Dark septate endophytes. This is sort of a thing you might not have heard of. These are root-associated endophytic fungi. Endophytes live inside of plant tissue. All plants are permeated throughout their entirety by hundreds of trillions of fungi. It's sort of a side topic. But these fungi are only found in roots. Interestingly, they only associate, or they can associate rather, with the few plants that don't form true mycorrhizal associations, like our brassica plants. So they actually might be helping be sort of performing that mycorrhizal role while not being true mycorrhizae. We can cultivate them, we can inoculate seeds with these types of fungi, but studies are very limited at this point about what the benefit is, and even potentially negative, blowback from doing this. So just always being cautious with new practices, but interesting to know about. These are fungi that are found in those cryptobiotic crust communities and desert systems shown on the bottom, and they've actually been argued to help translate or translocate some of the fixed nitrogen that's created in these crusts to the other plants in the environment. This is known as a fungal loop hypothesis, and you can look that up on Wikipedia. There's an article there. Also in soils, we get other pathogens and other bad fungi, if you will. I consider these the vocal fungi. I think in many ways this is just a slight perception change and paradigm shift, where when we see these fungi doing quote-unquote bad things and to our crops and woodlands, I think the question we need to ask is, what did we do to bring this about as humans? There's a great book called The Triumph of the Fungi by the mycologist Nicholas Money that sort of points out and lays the facts that most of the great fungal blights of history were caused by human mismanagement of an ecosystem. Of course, monoculturing sort of being an extreme example of that, where we strip away endophytic and mycorrhizal diversity, the plants are left defenseless, and sure enough, something comes in to sort of reset the scales. These fungi are ever present, but why don't they win out in the woods everywhere? I think it's because we don't have the right populations, perhaps of certain types of fungi, and also because sometimes, of course, we accidentally, as humans, import a fungus and it just wins the battle because it has no natural competitors. So, of course, that can happen. Fungi are integral to nutrient cycling and all these roles. On the phosphorus end, they can break down organic matter, decompose it through their phosphates from normal organic sources. With their strong acids, as I mentioned, they can release inorganic phosphorus from clays and also mineral phosphates. We are, of course, facing peak phosphorus in the future, so reducing our extraction of phosphorus from unsustainable sources is going to be important. Bringing these mycorrhizal fungi and other microbes to help naturally do this is going to be really important, and is now and certainly into the future. For the carbon cycle, decomposing fungi provide almost 98% of the carbon dioxide needed for the vegetation of the world. Humans and other animals provide, about 2% of the rest. About a third of all fungal biomass is carbon, and much of that is stored in soils. Again, with the glomalin, that's a major soil carbon sink that we need to be aware of in our studies. And that might be one of the primary, or might be even the primary, it's been argued, carbon source for whole soil microbial communities and fluorescent pseudomonads, nomads. That's a huge ecological impact that is often very much overlooked. So what I'm showing on the bottom is plants photosynthesize and trap CO2, turn it into sugars, which translates into mycelium, and even mushrooms and truffles that animals eat to spread around that carbon. The nitrogen cycle, fungi help release it through, of course, normal decomposition and organic matter release. Many ectomycorrhizal mushrooms will scavenge for ammonium. They don't usually prefer nitrate, so they'll actually help sort of gather that for the plants, which also prefer ammonium, generally speaking, so they can help gather that. They tend to dominate in more nutrient or nitrogen poor soils, whereas the arbuscular mycorrhizae tend to predominate in phosphorus poor soils, something to be aware of. And as I mentioned a little bit ago, some species like the Lacari mushrooms actually can eat insects. And in one study, they found up to 25% of the nitrogen in a Lacari-associated tree was actually provided from these insects. So even just the consumption of the killing, or whatever, if you will, of these insects can be a major nitrogen source for plants. Arbuscular mycorrhizae actually significantly contribute to the nodulation and association of rhizobia, nitrogen-fixing bacteria, on leguminous plants. They actually work synergistically in a tripartite relationship. So when we're talking about rhizobia and nitrogen fixers, we need to think about the AM fungi. They can work together sort of synergistically with infection and mineral nutrition in the plant growth. On one hand, the fungus provides phosphorus, carbon, and zinc that it sort of brings in through its network, which assists in the nodulation and nitrogen fixation process. And in exchange, the bacteria enhances the phosphorus uptake in the fungus and the plant with its own organic acids and phosphatases, digestive enzymes. The bacteria seem to influence the spore germination, even the growth rate of the fungus. At the same time, the fungus releases what's known as the Myc factor, MYC, and that helps activate the rhizobia formation or the nodulation. And together they form a beneficial biofilm in what we call the mycorrhizosphere, the fungal root interface. So they're important there. And so whenever we're bringing in the the nitrogen fixers, we want to bring in the AM fungi. Lichens are actually an important part of soils. In the background, we see that forests start out from bare rock, and usually the first thing showing up on that bare rock are lichens. Just like in the early earth, the fungal acids produced by the 95% fungal component of a lichen releases those minerals, enables mosses to grow, eventually leading to the climax forest. In that forest, lichens can actually provide up to 50% of the nitrogen input just through their own nitrogen fixation. In the lower left, we see one of the darker types of lichens that are primarily photosynthesizing due to cyanobacteria. Cyanobacteria can fix nitrogen. So when that lichen falls and decomposes, it releases that to the soil along with the minerals that it collects out of the atmosphere. Probably a big mineral input into soils is due to lichens kind of trapping that out of the atmosphere and falling down and releasing that when they decompose. And even the brighter green lichens in the upper right, like the Loberia pulmonaria, lungwort, probably needs and has a nitrogen-fixing bacteria as a part of its ecosystem. Lichens are kind of like micro-ecosystems, so it's also fixing nitrogen, and when it falls and decomposes, it provides that to the soil. So up to 50%. Huge input. Nitrogen-fixing bacteria do it. Lightning does it. Lichens also do it as a big part. And lastly, we'll talk about the remediated roles of fungi and sort of their healing capacities. Sort of simply and shortly put, there's two main ways that fungi can help deal with polluted soils or help with polluted soils. On one hand is with the heavy metals, which of course are elements on the periodic table that can't be turned into something else. They are what they are. All fungi can do is either move them around or sort of lock them up or bind them up so that they don't harm or interfere with other organisms. Just like plants, many mushrooms can accumulate these metals out of the soil and concentrate them into their fruit body. The question then becomes, what do we do with the heavy metal mushroom? Just like the question with sunflowers is, what do we do with the heavy metal sunflower? Because it can accumulate a lot as well. Unfortunately, we don't have a good answer to that on the backyard scale. Industrially, they would incinerate the plant or mushroom in a way to trap the metals in the ashes. So that is an option, but not accessible for most folks. Generally, we have to just dispose of it as toxic material, unfortunately. But at least we are scrubbing the soil slowly. And this can take generations. It's not a one-season process. In the upper right, we're seeing these Lixinum mushrooms still accumulating radioactive cesium outside Chernobyl, almost 40 years later. So it's a very slow generational process, but fungi do it. And if humans weren't there, the bugs would eat the insect and very slowly over the eons redistribute those metals into trace amounts as best they can. In the soil, fungi can also release acids and other compounds, even glomalin, which can bind with metals and sort of stabilize them for an unknown amount of time. They sort of just stay there in complexes, but later on, other fungi can actually come and re-release those minerals, as we talked about. So it's heavy metal contaminated soils are difficult, and we can't really do much about them and control them. That's why industrially they just scrape the stuff up or they burn it. Not awesome, not great options, but it's where we're sort of at. Lastly, and perhaps more interestingly, some fungal components on their tissue can actually bind to metals naturally. And when we put these the mycelium into, say, especially like water systems contaminated with metals, the metals might bind to the mycelium and sort of pull it out in that way. And that's an interesting application that I think is more accessible to folks. Lastly, fungi can break down all kinds of chemicals. And this has been well researched for something like 50 years. By many researchers around the world, initially starting out with wood decomposing fungi. Again, these fungi are able to break down the most complex thing in nature. So some smart person a bunch of years ago said, hey, maybe they can break down all the complex chemicals we're inventing as humans. Turns out wood decomposers can break down many of these chemicals, but so can many molds. The Trichoderma molds I mentioned can break down glyphosate, meaning Roundup. They can break down DDT. Many other herbicides naturally, so even simple molds can do this work. Many ectomycorrhizal mushrooms and even arbuscular mycorrhizal fungi can break down many chemicals. So it seems to be a natural trait of many fungi. Even fungi harvested from pristine environments can break down herbicides naturally. The challenge now that we face as people knowledgeable on this topic is how do we translate what we've learned in the lab, where most of this work has been done, into field-based applications and protocols that are consistent, safe, and measurable. And that's sort of the challenge today. Much of my work is trying to advocate for that research and enable people to do it safely and intelligently with good design. And then in the upper right, we're showing that different types of fungi can break down even plastics. The fungus there is actually a wood decomposer, not a soil fungus, but for the 100 years of plastics existence, many studies have been done around the world where they've buried plastics in the soil to see how they would stand up. And time again, every single time they dig up the plastic a year or two later, it's being degraded, slowly broken down, especially by fungi, sometimes even distinct clusters of fungal species. So here again, we know this, yet it has been translated to a larger practice. Hopefully in the future we will dial this in and deal with this major issue. So fungi are pretty, they do so much in our environments. There's actually many ways we can incorporate them into our designs as, say, permaculturalists, if that's your leaning, or just, good farming practices, or even just trying to have a garden in your backyard. Fungi can be incorporated in a myriad of ways. So often when I read books on these types of topics, one of the first things I'll do is flip to the index and see how many entries are there for fungi, mycelium, mushrooms. And more often than not, there's none or maybe just a couple, and they're very cursory. And I point that out just to say, not that nobody knows anything. I mean, that is sort of an issue, but more to challenge you to always ask yourself, how can fungi fit into this design? Even if the book or whoever you're talking to hasn't said it explicitly because it's not necessarily on their radar, now it's on yours. And you can start to think, what are fungi doing in the system? What's missing from this analysis? How could fungi be incorporated? And ultimately, cultivating fungi and working with them isn't too complex. Biology is unfathomably complex, but at the same time, it's relatively simple. A seed puts in the soil and it sprouts. Fungi are kind of straightforward in the same way. Once you understand them, it doesn't have to be a scary thing. It's just our language and our dialogues need to be a little bit more on the same page with each other so we can start to advance these conversations. So hopefully this talk gave you a good whirlwind introduction to the soil fungal communities and all the things they do. So I want to thank you all for joining in this talk, and I'm really excited to dip into the questions. Last thing I'll say before we do that is as a thank you for watching this talk, you can, I'm offering a discount on the sell of my book. It's at the website below, catheus.com, and if you enter the coupon code SOIL, when you check out, you get $5 off the book for the next 24 hours. So that's going on, and otherwise you can check out radicalmycology.com for some free videos. There's a lot of free resources there, and we're on social media. Rad Mycology is the handle. So with all that being said, I guess we'll turn it back over to Raleigh, and I guess we'll open it up for questions. So thanks, everybody.
Man, that was fantastic, man. It's totally a fire hose of amazing info. Like the stuff on glomalin, that blew my mind. I had no idea that was the biggest source of carbon. Great. All right, so for anyone else around more about Peter, you can find out more about him on radicalmycology.com, which is great. I posted the link down below. And one of you guys, we're going to announce in the webinar replay, which we'll send out over the weekend, one of you guys is going to get a free copy of Radical Mycology. So, how cool is that? Yay. How about enter one into the chat box if you thought if the presentation was fantastic, because I know I had a fantastic time learning from it. Yeah, tons of ones coming in. One. Fantastic. Cool. All right, so without further ado, let's get to some Q&As. I know a lot of you guys are anxious. We've got about almost 250 people here, so there's going to be a lot of questions. So try to keep it to your most important question, and we'll get to it. So here we go. Starting out, Q&As. Who do we have first? Okay, so starting out, first question is from Lauren. If we take a core sample from a healthy forest, can that be used to inoculate a forest garden?
Potentially. So what I would do, if you really want to be specific about it, and you don't necessarily have to do all these steps, but to get the best understanding of what's going on, is you can take that soil sample, say 12 plus inches deep, different fungi living, of course, at different layers, and on the arbuscular mycorrhizal end, like I showed in that slide, you can sieve the soil and actually separate out the spores, look at them under a dissecting scope, and quantify their diversity and abundance. And then use that to create and amplify those spores and create basically more mycorrhizal inoculum of those locally adapted species and strains, bring them back into your soils, and then in a really good practice, maybe a year or two later, sample your own soil. Of course, doing it at the beginning and then doing it a year or two later after you inoculate and seeing how that population has changed. That would be, give you the most information. If you want to be a lot more passive about it, then yes, you can just take that soil, ideally in the winter months or sort of when the trees have started to shut down, and that's when a lot of energy goes into the roots and the plant stops photosynthesizing, and that's when the fungus sporulates in the soil. So that's when you get the most spores. Sample the soil. Again, just you can cheaply or simply inoculate grasses in a pot or bag without necessarily knowing how many spores are in there, and very likely there's some stuff in there, some spores in there. Cultivate the seeds following the Rodale protocol I mentioned. They actually talk about using wild soils in that protocol. And then, ideally everything goes well, and the next season you harvest that mycelium and spread it across the land. You're going to want to amplify it. Just taking one sample, you don't know how many spores are there, trying to spread out throughout a whole forest garden. You'd be better served to spend the year amplifying it up.
Cool. All right, so next question from Birgit. I actually want to ask this too. How do you encourage beneficial fungi in your soils? Like practical steps. Let's say you're doing an agroforestry thing in the backyard and you have wood chips. How do you actively encourage more fungi to grow?
Well, it depends on what kind of fungi you're talking about. Decomposers are just going to naturally show up on that wood. Again, fungi are the only things that can break down lignin, so they'll eventually get there. But wood chip piles can take a really long time to turn into what we would call soil. So we can speed that up by inoculating those wood chips, and that's where getting into cultivation awareness and skills, which as I say, they're new to most folks, but once you learn them, it's relatively straightforward once you understand the theory and practice. And so, you can inoculate those chips, benefit them that way. That could be an imported culture. You can learn to grow local species. So it just kind of depends on the approach you want to take. If you want to be passive, then let those chips sit, and they'll break down over a bunch of years, and fungi were involved in that. If you want to bring in or amplify or support mycorrhizal fungi, our best practice is to try to reduce our impacts on soil. Tillage, chemical inputs, both these things really disturb fungi. The tillage, of course, breaks up the mycorrhizal networks or the mycelial networks and also churns their spores deeper where they don't normally go, so that's not good for them. When you over-fertilize a plant, as I said earlier, the plant doesn't need the fungus, so it'll decouple. And of course, chemicals and fungicides and pesticides also impact fungi. Some of them can survive it, break it down as I talked about, but it's not necessarily we want to do that intentionally or try to stress them out. So, sort of going back to the no-till practices, Fukuoka type of approaches are just letting the soil be soil and do its thing and let it all build up. Once those populations get in balance, then things start to adjust, and that's why his practices worked, because he was just allowing the fungi to grow. I think that was a big part of it. We don't have those fungal communities off the bat usually because we've done so much to destroy our soils. So that's often why the things turn all moldy and become problems. And we're like, well, this no tillage is a bad idea. Well, it's because you don't have the beneficial fungi there. And so you need to wait a few years and let them come back, or try to inoculate that soil to speed up nature's processes so that it can then do what it naturally does in a healthy forest. So then it gets beyond, like I say, if you want to say grow a garden mushroom for food on your wood chips or other egg waste or your sweet straw or something like that. There's a lot of good practices, 100-year-old practices. We can do that to build our soil, bring in another crop, but it's extra skills, not necessarily passive and simple.
Awesome. Okay, next question is from our good friend, the permaculture chef, Seth Peterson. He's asking, he wants to know, he's in Hawaii right now in a tropical climate. He wants to know about remediation since there's lots of pesticide use on the islands, and agroecology and Hawaiian fungi in general, if you know about those.
Well, I don't know much about Hawaiian fungi. I actually just read a paper about something about Hawaiian fungi the other day, and I can't really remember what it said. So you can't speak about them too much. So the question is how to deal with pesticide-laden soils, I guess, and bringing fungi into that situation. Well, when we're talking about really broad-scale remediation efforts, especially in that type of situation, or say like a brown field, even more extreme, where you just have no idea what happened to the soil for centuries or decades in an old factory, we don't have these dialed-in protocols. I mean, what we can do is, on the more passive end, try to see what fungi are there, what plants are there as well, because plants are doing things to help the soils, bacteria, and we can bring in some of these more natural or sort of simplistic approaches of phytoremediation, bringing in compost tea, aerating the soil, this type of stuff, and of course, inoculating fungi, bringing them in, trying to recreate a healthy ecosystem. Which fungus is the best, which blend of species, we don't have these answers at this point in our very, young age of mycology and mycoremediation. Certainly, I think the first step is observing and seeing what's already there, amplifying those indigenous mycorrhizal fungi that are tolerant of those chemicals, if not, engaging with them, breaking them down. And if you're testing the soil and not finding any fungi, well, then you work with what's the local strain maybe and try to bring that in and see how it responds. This is one of those situations where I think chemical contaminated soil is actually probably one of the easier approaches, in quotations. If we're talking about acres of soil, then it gets kind of hard, but in a small patch, like an oil spill, like a small little 55-gallon drum you spill of a chemical, this is where we can actually dig up the soil, inoculate it with, say, a decomposing mushroom. It'll run through that, break down a lot of it, and actually do some pretty good work. And that's that really famous Paul Stamets anecdote where he did this at a bus depot. And that kind of situation where it's more controlled, contained, it's just a stable, solid material that a decomposer can quickly grow through, that's actually pretty approachable. But when we're talking about acres and acres, we're essentially trying to recreate a healthy ecosystem in the long run. A big part of that is bringing in the fungi, which are probably absent or severely depressed or low in diversity. And again, I can't say which is the best species to bring in other than which ones are growing locally or which ones are already there. Ultimately, we've done these things that are pretty unnatural, and nature doesn't necessarily have, quick solutions to them, but we can try our best to speed up what it can do.
All right, next question from Elizabeth. She'd love to build on my excitement. She says, Wayne's excitement. I'm actually Raleigh, Latham. Thanks to Wayne Dorbin, he lets us use the software. But anyway, she wants to build on the excitement about glomalin. She says, can Peter say more about the potential of fungi to help sequester carbon? Good question.
Yeah, I mean, it's huge, right? Huge concept that I was really trying to emphasize is very much overlooked, especially when we consider that glomalin and mycelium and other exudates might be the primary carbon source for soil microbes and insects. It's huge. And how often do you hear that? So, basically what I said, I can't really say too much more. Trying to just be aware that this is an integral, incredibly important part of our soils, of all of our ecosystems fundamentally, and we need to discuss it, amplify it, work with it, bring them in, support their growth, not destroy them. And of course, when we're planting trees and doing all these other things to sequester carbon, we need to bring in the fungal communities and really perhaps, support the plant mostly so it can support the fungus. I think our perspective has been the other way around. We need to think about trying to support the plant so much. Really, if the fungus is happy, it's going to support the plant, right? And so that's kind of as much as I can say. And then doing it, using less chemicals as another source of nutrient waste, because these are also going to impact fungi.
Okay, cool. Big question from Baba Bear. This is one, it was about Paul Stamets and Elaine Ingham. He's got a very specific question. He says, Paul Stamets mentions adding gypsum to grow fungi indoors. Elaine Ingham, on the other hand, says that using any type of lime and gypsum kills the microbiology. It is a salt, so it harms it. Can you speak a bit more about this?
Well, in indoor cultivation, we use gypsum as like a sulfur and calcium source. It's calcium sulfate, but mostly we use it as a flocculant. It makes the materials we grow mushrooms on more fluffy, so flocculants sort of push things apart, and that's good for growing mushrooms. It's easier for the mycelium to grow through the sawdust particles or whatever we're growing it on. It gives it a more structural quality that's just better for growing. And that's primarily what I use it. It's a little bit of a mineral source, but mostly it just adds to water holding and structural qualities. It's actually news to me that it's bad for microbial life. That's actually really good to know for outdoor. That's great. That's kind of as much as I can say. So yeah.
There we go. And Antia says, any info on fungi and nuclear waste?
Yeah, so nuclear fallout is usually radioactive isotopes of plutonium, uranium, but really commonly cesium. That's what hit us in Fukushima and Chernobyl, cesium-137. So these are elements on the periodic table that can't be turned into anything else. They can't be broken down. Just like other heavy metals, they can be bound up or moved around by fungi. But they're still probably going to be releasing the gamma radiation that interferes with and mutates our DNA and causes all the problems. So fungi can't stop radiation. This is a sort of a misconception that I come across commonly, and it's totally understandable. All they can do is accumulate those metals, especially if they're a mushroom former. So in that slide, if you remember, I was showing those Lexinum mushrooms from Chernobyl. 40 years later, they're still radioactive. That was a Geiger counter showing them being radioactive, because they're pulling up the cesium, but they're still emitting that gamma radiation, so that you can't eat those mushrooms. Deer actually eat them. They get cancer. They leave the exclusion zone. Humans eat those deer. They're getting cancer. This is actually well known. So they can't stop it. All they can do is slowly pull it up. And so when we think about, well, let's inoculate the west coast of the U.S. or Japan with a mushroom that is known to accumulate radioactive cesium, what's going to happen is over probably generations, probably maybe even hundreds of years, maybe who knows how long, we don't even know, the Japanese people or the West Coast folks will have to harvest these radioactive mushrooms, theoretically in protective suits, and dispose of them as toxic radioactive waste, because that's essentially what they are. We can try to burn the mushroom and concentrate it in this really controlled way, as I mentioned, and even put it into glass. That's known as vitrification. Not very easy to do in the backyard. So we have to be aware of this. And I didn't mention with the metals that really what the problem is that most of these metals, these toxic metals, are often deep in the earth and only released through like vulcanization or erosion sometimes. We as humans extract them and naturally concentrate them and then accidentally spill them. Nature doesn't have a way to deal with this very unnatural system. All it can do is very slowly accumulate it, and like I mentioned, a bug will bite the mushroom, go over here and deposit a small trace amount of it, and over centuries, millennia perhaps, it gets redistributed to trace amounts and slowly works its way back down the soil. But it's a challenge, certainly. And so for me, the best medicine is preventative, and so perhaps we need to look at alternative energy sources rather than trying to bandage this huge issue.
All right. Brendan's asking, is there any fungal tea recipes for adding fungal life?
Yeah, like compost tea. It's a question I get pretty often, and from what I am aware of, and folks out there might be able to correct me if I'm wrong, but most compost tea are cultivating some fungi. You're probably cultivating some yeast, probably some mold to some degree, but you're very much likely not cultivating a mycorrhizal fungi. These generally need to be in association with the plant to live at all. AM fungi, Arakoid mycorrhizal fungi cannot live without being in association with the plant. Even in the lab, we have to inoculate carrot roots and study the carrot root and fungus intersection because we can't grow it on its own. We can grow like chanterelle mycelium on its own in the lab, but under very controlled conditions, and it's a little bit more challenging. But it needs very specific minerals and salts and things like this. And we're not getting that in simple molasses, tea, water, that type of thing. So maybe some molds, probably some yeast, and there's benefit to those things, as I mentioned. What's the best fungal tea? That's not my area of research. From what I've heard, people looking into these types of topics have actually struggled making a good fungal tea. That's what I've been told by people who looked in this much more deeply. And perhaps in the future we'll come up with some, killer fungal tea recipe. But as far as I'm aware, there's not a good one at this point.
All right, Jacob asks, what is a good way to balance fungi for tree and soil support versus human consumption?
Well, I'm not actually sure how to mention that or answer that. I mean, generally speaking, if we're bringing in fungi to support the trees, we're going to get benefits probably from the trees that we're trying to grow and the soil and all these things. The tree needs to be healthy. And a lot of our trees need the ectomycorrhizal fungi to survive. I guess maybe one thing I think about with that question is perhaps hopefully in the future we can inoculate all of our trees, all of our productive trees and just woodland trees, what have you, with fungi or with mushrooms, Ectomycorrhizae, that produce edible and medicinal mushrooms and truffles. Right now, like I said, we can't grow chanterelles, we can't grow boletes or matsutake or morels or something consistently in this way that we wish we could, probably because we're not mimicking the soil conditions properly or even the whole plant community that the plant fungus needs. Usually the studies are working with sterile soils, so very unnatural, and of course the mushrooms aren't forming in the lab. So we just got to figure that out, probably bring in some compost tea or who knows what else, some other shrubs nearby to get the mushroom to fruit. Right now probably one approach is to bring in Leucaria mushrooms. Those are very beneficial to a lot of trees, and they're actually edible and medicinal. They're not gourmet, but it's another crop in your rows of trees or what have you. So that's one approach. And then as far as balancing, you're not going to do harm by inoculating, say, wood chip piles next to your tree. The decomposing mushroom isn't going to attack a living, healthy tree, especially if it's not a mushroom that's known to be a parasite. I wouldn't necessarily inoculate my garden with armillaria mushrooms. These are decomposers, but also parasites. They can attack living trees, usually a sickened tree, a damaged tree. But, barring that species, most of our common edible cultivated mushrooms are totally fine right at the base of your tree. Probably not right next to the bark, at the base, but you're not going to be concerned about that really at all.
All right, another question come in. Ton of questions. Oh, here we go. From Jeffrey, can we inoculate to break down glyphosate in the urban gardens or farms in general?
Man, don't we all want to do that? And what's the quickest way to deal with all this, all this Roundup with fungi? We know, like I said, Trichoderma molds can break down. Trichodermas are amazing. They're incredibly common, easy to grow, nasty-looking green little things, but they do a lot of good work in the world. And certainly, I'm interested in that question: how do we just throw some Trichoderma at our soils and deal with the glyphosate? Sounds so simple, and maybe that is too simple, too simplistic a way to think about it, but maybe not. Maybe we can come up with a good approach to deal with it in this way. This is really where we're at, where there's these types of questions. This type of experiment would actually not be too hard. You know what chemical you're looking for, which is often the hardest part when you're dealing with chemical treated soils. So we can test for the concentration of glyphosate at a lab, cost a little bit of money, inoculate with Trichoderma, try 5 different types of inoculation methods, and then a couple weeks or months later test it again and see how it's changed. It's a pretty simple project. it's on my bucket list, but maybe you'll get there before me, and I'd be, happy for it. This is just where we're at, where there's just not a lot of people doing this work. Even these relatively straightforward things you think we should be looking at, or dealing with our plastics, right? But nobody's doing it, or at least not on the scales that we think it should be done, or at least I think it should be done. So this is so much, again, of what I try to do, and through my book, trying to teach people, how to cultivate, flash it quickly. So the biggest chapter is the cultivation chapter. There's a whole chapter on remediation, breaking down the sort of science and what to ask yourself. But I think once you wrap your head around it, it is relatively straightforward. It's a series of good questions and good design. And this type of experiment, certainly an important one. I haven't gotten to it yet, and I'll be happy if somebody gets there before me, that type of thing.
So Lena says she hears a lot of talk in her Mediterranean climate about reestablishing fungal ecology in the soil and how that's going to make a huge difference. Trying to get to the question. If we introduce lost soil, bacteria and fungus here, is that what will heal the nutrients cycle and result in better range growth? Your thought on that issue? It was a pretty long question.
Kind of as I've been saying, that yes, I think that in our depleted soils, we are missing the natural communities of fungi and microbes and insects that has led to the health of our planet before, humans did a bunch of stuff to sort of set it back. So we need to speed that back up, recreate it, try to help regenerate it. We can never restore, we can never go back, but we can try to develop best practices, holistic management practices where we're thinking holistically, thinking forward, going slowly, but also observing and trying to recreate what nature is already doing. And these are pretty fundamental permaculture practices. And again, just bringing fungi into that analysis, that picture is a part of it, bacteria, all these things. And so I guess the answer is yes. Yes, I think if we bring in more fungi, more microbes to our soils, and recreate that ecology, good things will come. If the minerals are all gone, or something like this, the fungi just can't magically create minerals. So we might need to bring in a mineral source or something like that, which is necessary for life. So there's other factors, but these microbes are a major component in releasing and moving and making available these nutrients for life.
Cool. As a follow-up to that, she wanted to know, is there specific mycorrhizal products for ag and garden use that you could bring in?
There are lots of products. I can't necessarily say one is better than the other. I know a number of people who've done as college experiments or projects in class. They'll look under the scope and actually test for try to quantify the number of mycorrhizal spores in 10 different products. And several times actually, I've met people from different schools did the same project, and they said that in some products they found literally 0 spores. So I don't know what products those were, honestly, but some are probably better than others. One that I use, but I haven't personally looked into the scope to quantify the concentration, which is one of the things I just need to do pretty soon, is a company called Bioorganics. They got a bunch of good species in there, a bunch of ecto and endo Mycorrhizal species. Seems like they know what they're doing, and looks like a good product. I haven't quantified it yet, but that's one that's out there. That being said, and I sort of alluded to this in the talk, is in one study I saw of a conservancy group in Washington. They were trying to amplify some indigenous endangered prairie plants. In Washington, there's a really endemic wildflower plant populations in some of the small prairies we have left from the Native Americans burning the oak savannas or oak prairies. And they grow up these plants in the nursery, put them back out 5 years later, and they all die. So in a study, they brought in some commercial Mycorrhizal inoculum, grew the plant out, put it back out, and some of the plants actually did worse with the Mycorrhizal inoculum. So when I saw that they were going to recreate the study using the local fungi from that prairie rather than the commercial product, with the hopes, and I think the logical hope that it would be better for all the plants. Unfortunately, I never saw the follow-up. I would imagine it actually was better and more beneficial, and that's certainly an approach that I would advocate.
All right, Matt asks, he's in the middle of reforesting an area with a huge forest fire. Where could he go for some information on inoculating the seedlings?
Well, yeah, I'm not really. I don't know any of the resources specifically for planting out trees in a burned area. It's not actually something I've never looked into. I don't know if it actually requires specific practices. It's actually a totally new topic to me in this moment. But I would imagine that it's going to be pretty similar to what we talked about. You're going to need to bring in the ectomycorrhizal fungi, probably the Lacaria and Rhizopogon and Pistolaris species that are known to establish readily, and that many woodland trees, or pretty much all woodland trees, need to survive. So whether it's a burn soil or non-burn soil, I'd imagine that, the tree still needs those fungus. Whether you need to do other things to support the fungus in that soil condition is a good question I haven't thought of before. So I'm not quite sure. I can't really say in that regard.
Okay, science question from Spencer. When a spore germinates, does it need to become dikaryotic to form a relationship with plants?
Well, that's well, I don't want to get too technical for folks that are new for all this. The dikaryotic thing, meaning that the organism has two types of nuclei, which is unique to fungi, an extended dikaryotic state. That's mostly with ascomycetes and the basidiomycete mushrooms, where we're thinking about this. I don't want to get too technical with it. I think the answer to what you're getting at is for a mushroom for say a basidiomycete mushroom like Lacaria, it can drop one spore that only has one type of nucleus, so it's monokaryotic, and it will germinate, grow a mycelial network, and actually live healthy for perhaps years. It can just never reproduce until it finds a genetic mate, fuses with it, forms a dikaryotic state, and then it can actually produce offspring. Whether that monokaryotic network, it probably must be forming a mycorrhizal network with the tree. When does it get dikaryotic? That's actually a really good question now that I think about it in that regard with the Ectomycorrhizae. I've never seen anybody answer that. It might be the type of question we actually can't answer because it's like in the soil and it's so hard to study in the soil. Pretty interesting question. Actually, I don't think I have a conclusive answer for.
Okay. So, Marianne, we actually have a bunch of people, like 6 people asked about desert truffles. So, Mariana is asking, she's in a dry part of the country, Costa Rica, Guanacaste in the north of the country. It's getting very dry, a lot of water problems, desertification, government. The universities are looking for other options to provide food for the people. She wants to ask, where can she find more information about those desert truffles?
Yeah, pretty interesting topic, and actually, to be honest, was one that was totally new to me when I was researching my book. I came across this whole concept and cultural history of desert truffles. I don't live in this part of the world or that part of the world. Pretty fascinating, and yeah, then turns out they've studied them. They can cultivate them as long as the soil conditions are more sandy and appropriate. As with so many aspects of mycology, no real good resources on it. There's one textbook from Springer that's like, I don't know, $200-300 I want to get, called just Desert Truffles. I haven't read it yet. And it has some cultivation information in there. Really, though, I think it's going to be pretty straightforward. If you read my book, I get into it where I talk about how to cultivate Ectomycorrhizal mycelium, which is what these things are. They're Ectomycorrhizae. You can cultivate in the lab. You basically create little jelly beans out of the mycelium, and then that's what they commonly inoculate the trees with. I actually showed it in the slide. I didn't mention it. It was in the Pistolaris screen. They're called myco beads, so you can look that up. And so you're probably doing something similar with the Terfezia, the desert truffle mycelium, and then inoculating the Helianthemum or Cristaceae family, Rosa Rock family shrubs, and trying to get it to establish. And actually, there's some successes. So that's kind of as much as I can say, and I get into those more technical things in my book. Another desert mushroom you can look at is Padaxus Pistolaris, the stalked puffball. This is pretty common in desert systems, and actually one of the universities in Pakistan was able to cultivate this mushroom pretty much in pure sand and get it to fruit, and it's an edible mushroom. So Padaxus Pistolaris, or the stalked puffball.
Cool. All right, so we're getting a lot of questions about just common ways to just inoculate wood chip piles, because that's one thing at Versaland when I was there. was like, how do people just easily find mycorrhizal fungi that will, one, help them inoculate wood chip piles to put on the forest, and two, help them break those down?
Sorry, deal with that.
Don't worry.
Just a simple way to inoculate wood chips with what I always say, sort of really the beginner mushroom, and anybody out there that's familiar with growing mushrooms will know what I'm talking about. Your go-to garden mushroom, especially for the beginner, and pretty much everybody really in the world, is our friend the King Stropharia, or the garden giant. Of the 3,000 mushrooms we can consider gourmet and potentially cultivatable, only about 30 have really dialed in product practices, and only about 6 are grown industrially, and really 3 dominate the market: buttons, shiitake, and oysters. Among those 30 is our friend the garden giant, and it's just been selected by trial and error and people looking at every possible option for the last 100 years to be this awesome species that is edible, grows really large, really quickly, is very stress, heat, drought, microbial, competitor tolerant, just really aggressive and doesn't care. Will eat fresh wood chips all the way to your old funky straw and composted material, so a very wide diet, which is uncommon to a lot of mushrooms we grow. And and turns all that material into fresh topsoil in like a year or two. So it's this incredible garden mushroom, hence its name. Tastes great. You know, there's other Stropharias, other mushrooms we can grow in the garden. This one is just so dang easy and consistent and reliable and just grows itself. So that's the one I would suggest starting with. And, you know, really when you get your best practice into best practices, what I like to do is, even if it's a wetter environment, but especially in drier environments, dig down a couple inches, say 6-9 inches into the soil, create a little reservoir where water can collect to keep everything hydrated. Mushrooms need a lot of water, so we don't want to pile the chips on top of the soil because it'll just run off and dry off more readily. Dig down. If you have good drainage, hopefully not too clay or compacted soils, I would line the bottom with burlap or punctured perforated cardboard just to provide some drainage, but also a buffer from competitor soil fungi. And basically create a lasagna out of, this is the simplest approach, lasagna out of fresh wood chips, like 2 to 3 inches of fresh, ideally hardwood chips is the best. And then sprinkling of the mushroom mycelium, or what we call spawn, which you can buy Stropharia spawn from pretty much any mushroom grower. It's so common and popular. You can get it from pretty much anybody. One company that I like, I don't work for them or anything, is Field and Forest, good family company in Wisconsin, provide good quality stuff. So you can get spawn from them. And you just lasagna, say 2-3 inches of chips, a nice little sprinkling, like a nice salt and pepper of the spawn, not too dense or nothing, 2-3 inches of chips, and just work a couple layers up till you reach about 9-inch deep of a bed. You could kind of mix that stuff up as you go to sort of distribute the mycelium. You even just kind of mix it all together on a tarp beforehand. You don't necessarily have to lasagna it. Same concept, mix it together. And then top dress it with about 6 inches of straw to reduce evaporation, basically your mulch. Or if you don't got straw right off the bat, just some cardboard to reduce evaporation. Once the mushrooms start fruiting, which can be in just a few months, it grows so fast, you'd want to replace that cardboard with straw so the mushrooms aren't constricted when they try to grow upward. Of course, we want to place that in a shady area, you know, on the north side of a building, under a downspout, somewhere it can stay hydrated, not too hot. Even though this mushroom's tolerant of heat and drought, we don't want to stress it. We want to just have it grow happy and pop out a lot of fruit bodies. Yep, it's a good one. It's a great one. Once you establish one patch of mycelium, you can break that up and spread it out through your whole garden and really get as creative and just throw it everywhere, feed it everything, and it's pretty fun to play with.
Yeah, that's great. That was one thing. Grant Schultz had a video about it where he took King Stropharia, he stuck it into his giant pile of wood chips that were taken from trimmings, and then he had a mulch spreader which he spread along the trees he planted. And then like 2 years later, I was just walking around and there's just all these different sorts of mushrooms, and those trees were super healthy compared to the ones that didn't have it. So it was really cool to see that in practice. Mm-hmm. So Uta Bonsack, coming on back, good to see it. He asks, do you have any experience growing edible mushrooms on shredded miscanthus?
I do not. I'm not even familiar with miscanthus, so you'll have to forgive me. What I can say, and I'm guessing it's some agricultural waste, I'm not sure what the crop is or even what it looks like. What I can say is that whatever it is, we can probably grow mushrooms on it. 80 to 90% of what we grow as plants is wasted. So we mostly harvest the seed or the fruit. The stalks and everything else is burned or composted or what have you. All that stuff can be turned into mushrooms. So much food that we're missing out on, really, and I didn't even touch on that in the talk. And then that turns into soil, good compost. It's its own type of compost. It's a solid state fermentation rather than the aqueous fermentation that we call normal bacterial composting. So it's another way to break down and release nutrients and increase nutrient viability. So all that being said, you could take whatever your plant material is, generally speaking, and grow mushrooms on it. You might not be able to grow it on its own or on that plant its own. You might need to mix it with other materials to get the right nutrient balance, carbon and nitrogen, the right qualities of fluff, and not too anaerobic or something like this. So these are considerations. Maybe minerals need to be brought in to supplement. But certainly that waste product could be incorporated into what we call a substrate formula, a recipe for growing mushrooms on. A lot of work been done in this regard for 100 years, growing mushrooms on all kinds of stuff, all the waste around the world. We know a lot about this. This crop I'm not familiar with, but yeah, I mean, I bet some money we can grow, say, our oyster mushrooms on it. It's a really aggressive mushroom, grows on all kinds of stuff, hundreds of ag wastes already.
Great. I'm trying to find a good question. There's a really good question somebody asked. It was about, introducing species. I'll find it again, but here's one from Lewis. Here we go. Another one from Baba. What actually makes a mycelium mat decide to come out as a mushroom? Is it the need to spread its spores, or is there a connection with the plants and other organisms it's connected to?
Well, this is, by my expression, this is actually a huge topic that I am fascinated by. Simply put, we don't know how the fungus communicates with itself to turn the mycelium into a mushroom. Unlike plant and animal development, where there's a lot of hormones and chemical signals known to be associated with organ development, there's no chemical associated with the transformation, the metamorphosis of mycelium into a highly structured and complex fruit body we call a mushroom. Arguably, it's just, to spread spores, but how does it even do that? In my book, I argue with a lot of information to support it that it's electrical signaling, whether electromagnetic or just internal electrical signals. There's a lot of evidence to support that. Even more strikingly, I think, to support that is that there's been a lot of good studies in China and Japan showing that when we strike a mycelial block of a block of mycelium, which is what we fruit mushrooms from on the farm, with like 50,000 volts for 10 nanoseconds, much like a lightning strike, we will consistently double or triple our yields. And they can do the same thing in the forest. They can actually, they've done this in studies, they'll shock the forest with basically a fake lightning strike, and they'll increase the production of ectomycorrhizal mushrooms. So there's definitely an influence on fungi with electricity, and actually I don't think it's just circumstantial. I think it's my guess that it's a part of their communication. We just haven't looked at that at all, and I think in the future we'll probably be able to understand this more, be able to grow mushrooms better, and all kinds of things like this. So it's actually a big mystery, a really interesting and kind of crazy topic. But fundamentally, we don't know how they fruit. Basically, what we do in the farm is we grow mycelium. That's very easy. To get the mycelium to turn itself into mushrooms, we can't like sprinkle a chemical on it or something. We basically just try to recreate the conditions it wants. So we drop the temperature, we increase the fresh air and the humidity, recreate the fall essentially, and that's what triggers the mushroom to fruit. But it does it itself. And I guess just to follow up, in the forest soil with Ectomycorrhizae, it's even more complex. Is it what are the signals going on there? It's like I said before, with like chanterelles in our backyard. It's more than just it needs soil in a tree. It probably needs other insects and a whole plant community and all kinds of dynamics we've probably yet to understand. Maybe some sort of electrical signals or a lightning strike or who knows. But it's probably multidimensional. Hopefully not too complex that we'll never figure it out. But we haven't figured it out yet.
Okay, Lane is asking, is there any Native American varieties here in North America of the desert truffle?
It's actually a question that I don't know. I'm not familiar with it being like a traditional food source or food crop or any indigenous cultures in North America. I haven't had a chance to look into it. Again, resources are limited. Actually, probably a resource I could look into, and this just isn't an area that I've touched on. Maybe field guides from like Arizona. I might mention something. I haven't actually looked into that specifically. I do know that the Padaxus Pistolaris, the stalk puffball, does grow here. It's not a truffle, but I think that we could potentially grow it in North America if it doesn't already exist. On the West Coast, we have like our Oregon truffles. We have other forest woodland truffles. These are common edibles, gourmet edibles. The desert ones, not commonly talked about. I haven't come across it. Most of the research and documentation I've ever heard was from, North Africa, the Middle East. I would have imagined that reading all these things, there would have been a mention that they also grow in North America, and I never came across that.
Cool. Gabriel asks, she says, great presentation. When you're trying to identify fungi typical living in local soils, what's the basic process if you are not seeing fruiting bodies? And does your book have profiles of different fungi to help identify species?
Sorry, can you say the first part of the question again? How do you identify? Yeah.
What's the basic process to identify fruiting bodies, and how do you identify different types of fungi fruiting body species? Sorry, Gabriel, I may have slaughtered your question. It was a long question.
Yeah, well, I mean, it's a pretty good question. How do we identify mushrooms safely? It's a very large topic. Certainly, we want to be safe when we do that. There are poisonous and deadly mushrooms. Never want to forget that. They're very much in the minority. So don't, there's more poisonous than deadly poisonous plants than there are deadly poisonous mushrooms. So just also being aware of that. The best route really is to join your local mycological society. They're going to lower that learning curve so dramatically more quickly than any book can. You can piece it together by going out and learning from a field guide. There's a lot of good books out there that'll walk you through the steps, but it's intimidating. You're always second guessing yourself as a beginner. Having a mentor, an elder to guide you through it is very important, very helpful. So I always recommend that. When you actually get into it, it's pretty straightforward. You go out to the right habitat, right time of year, or you just go blindly and see what you find. I mean, that's just, whenever you go for a hike, you're also mushroom hunting is how I look at it. And you harvest what you find, and then you use a field guide, and the field guide usually works you through step by step, comparing photos and descriptors. And just like with IDing plants, everything needs to line up. Sometimes you might need to get into microscopic features. That's usually not necessary for our common popular edibles. For the guild mushrooms, what we call the agarics, it's very, very helpful to take a spore print, cut off the cap, put it on a piece of glass, and see what the color of the spores are. That will actually help you narrow in on the ID much faster. It won't give you this exact species or anything like that, but it'll get you a lot closer because the color of the spores will help, clarify what, it'll narrow in your options. That's like a first step, but then it just gets into pattern recognition, all these types of things that an elder is going to just get under your belt a lot quicker.
All right. David asks, can you point to any good resources or replicable methods for small-scale projects cleaning diesel-soaked soils using oyster mushrooms? He said he was looking for something in journals and he couldn't find it.
Yep, and I mean, really, this is where we are at. It is this kind of perplexing question, that I have. Why we have a wealth of knowledge from the lab, in very small 1 liter containers. These types of studies where they say, yes, this fungus can break down this chemical in X amount of hours or weeks to maybe even 99.99% efficiency. And then they shelve this, they write the paper, and that's about it. They don't follow up to say, okay, now how do we actually deal with the spill of that chemical? And as far as I've seen, I don't think I've really seen any real peer-reviewed studies looking at that kind of thing. There's probably a lot done industrially. There's a lot of bioremediation firms. There's even some private mycoremediation firms around the world. But they got proprietary knowledge, even their own patents of techniques. And so these are limits, barriers to access, and even for economic incentives for people to pursue this as a career, because the knowledge is made unavailable in a variety of ways. So there's probably a lot known industrially, but how that's percolated out to the general populace, scientific communities, seems pretty limited as far as what I've been able to access. And even these very straightforward, important questions, a diesel spill. Diesel is not super complex as far as a petrochemical. And it's a relatively common thing that we spill. We should have a pretty good way of dealing with this. Why isn't more money and grant money going towards it? I don't know. That's why a lot of my approach has been grassroots and bottom up, and saying we can do it together because the work isn't that hard. You know, you got to raise some money to do the testing, to do it right and do it proper and have some numbers. But actually, the work itself is not too hard. You want to be safe, etc. So all that being said, I'm not really familiar of any good studies in that specific example. You know, the one that Stamets did with the Department of Transportation in Washington about 10 years ago has a paper out. They didn't have any conclusions. You know, his photos are pretty compelling, and it actually, you know, obviously was doing some pretty good stuff. But even in their study, they couldn't draw a conclusion based on some variables. So stuff gets tricky. Not to downplay that work or anything like that, even just in what's out there, it's pretty limited. And even that was a great study, but no good follow-ups, you know, and it seems like, why isn't every other transportation department doing that kind of thing, right? So like I say, I don't have answers to these pretty basic questions. So much of what I do is just saying, well, that's the state of the affairs. Let's move forward. Let's teach each other. Let's do it ourselves. Let's develop really good dialogue, educated and informed dialogue, so that we can actually have good conversations about this, actually know what we're talking about. There's a lot of misconceptions and, you know, misunderstanding around the topic because it's so new and confusing to folks. A lot of what I do is trying to just clarify that, and then we can move forward. And that's why I've taken a big step back from pushing micromediation, which I did years ago, because people couldn't have the conversation. I couldn't have the conversation. I actually, you know, I didn't know enough years ago either. But now I'm just trying to, you know, bring us all up to the same page, move forward, and then we can do this stuff. This is the thing. This is where I always want to take it. It's not a pessimistic world that I live in. It's an optimistic one, but only once we can have good dialogue.
Awesome. So Mikhail says, you mentioned the Rodale Institute on cultivating your own AM fungi. Have you done it without buying the Bahia grass seed, and can you do it on your own native grass?
Oh, yeah, absolutely. All grasses associate with Arbuscular mycorrhizae. You can also use, you know, alliums. You can use all kinds of plants. They chose the Bahia grass because it's quick growing, deep rooting, so a lot of surface area for the fungus. It also dies in, you know, the slightest cold. So there's no risk of it going to seed in their northeastern climate, and that's why they chose it. And you know, they don't want to be invasive, but you can use a local grass. What you don't want is for the plant to put a lot of energy into necessarily seed production. You want it to basically put a lot of energy into its growth and into its roots and things, and then so it dies. In that example, it dies when it gets cold. And then that shocks the fungus because it's like, oh, my plant partner died, I need to sporulate. And so it produces a ton of spores in the bag or the pot, and then you harvest the roots, the mycelium, and the spores the next season. If you want to use a local grass, just let it grow kind of to its maximum, and then right kind of like at the end of the height of summer, chop it down, kill the plant, shock the fungus, and you'll do essentially the same thing.
Great. So we're almost, we're going to run out of time soon. That's why some people are asking, hey, does Peters have any workshops coming up? And also, what was the link again for the book?
Yeah, so I didn't mention it earlier, but I'm actually going to be doing a workshop in Chicago this coming fall on soil fungi. And I'll be working with Nancy Clem. She is an amazing bioremediator, horticulturalist, ecological designer, soil wizard, compost wizard, and an amazing person. You should check her out. And we'll be collaborating on probably like a week-long, 5 to 7 days, I'm going to quite out in soil fungi intensive. So it's a whole new class I'm going to be offering. I'm pretty excited about it. Details aren't up yet, but it'll be fall of 2017. So basically just stay tuned to radicalmycology.com. If you go there right now, you can see links to the workshops I did this last season, and there's a sign up at the bottom of all those pages and at the top of some of them where you can enter your email. Any of those pages, I'll get your email, get you on my cultivation course list, and you'll hear about all those offerings once they're dialed in. So that'll be in Chicago this fall. I'll probably also be doing another tour this coming fall. I've done one in the last few years around the country, offering lots of different options. And I'll probably be doing a lot of new stuff. So I've been doing mostly just mushroom cultivation the last few years, certainly expanding out to a lot of new exciting topics I have yet to unveil. So stay tuned for all of that. And yeah, social media, of course, and following the website is going to be the easiest way to do all that. Our handle is Rad Mycology on all the common ones. And then, of course, there's the email list if you get on that. And then as far as the book, you can just go to Rad Mycology. So right at the top where it says RM book, kind of on the top bar, you click that link, it'll take you right to the page where you can order the book. The website is CHTHAEUS, Catheus.com, kind of a weird word. And if you buy the book in the next 24 hours or by tomorrow, and you enter the coupon code SOIL in capital letters, that will give you $5 off the book. So a little incentive, get you going for the holidays.
Nice, good stuff, man. Yeah, we'll definitely post that link in the replay. Everyone's getting a replay. It's going to be awesome. We're going to leave it open for free till Monday for folks who only dropped in for a little bit. Let's get back to a few more questions because, man, there's so many good ones, and it's tough having a great webinar like this because we had 250 people. No, at its peak we had 270 people show up, and so we have hundreds and hundreds of questions. But here's just a few more that we're going to do. So Mary asked, she doesn't have the names, but there's a couple of root rots that are causing a lot of pine trees to die in her area of northern Idaho and other places in the Pacific Northwest. She says, can I inoculate wood with some good guy fungi and scatter around in my woodlands to outcompete the bad guy fungus? If so, what species of fungi would you suggest?
Potentially, decomposing mushroom bed on the sort of top of the soil might through its decomposition and just natural growth release antibiotics and antifungals that would, by coincidence, outcompete or fight off some of these root pathogens. That being said, a better practice and probably what's more needed is to actually get some of the soil fungi into the ground, not necessarily mushroom formers, but our Mycorrhizal friends, our Trichoderma friends. These are the things, like I mentioned in the slideshow, have been well studied to fight off a lot of our common root rot problems. The thing about it is that we ideally need to get those fungi in the soil in association with the plant before the pathogen gets there, so it's preventative medicine. If it's already there, what are you going to do? It's certainly an effort you can make. Will it work? Not necessarily every time, especially if the plant's really sickened and there's a lot of problems going on to begin with. But that's where you're going to be looking. You're trying to get soil fungi in, not necessarily our fruit, mushroom-forming decomposers that live on the top surface.
All right, David asks, thanks for being so open source with your knowledge. What advice can you give to beginning cultivators?
Don't give up. Every new skill is tricky, and then you get better at it, and it gets funner, and it's always fun as long as you stay, happy while you're doing it. Don't do it in a way you're going to get stressed out and overwhelmed. Really common to get, really excited, but so much that you take on too much, trying to grow every mushroom in the world all at once. So don't do that. Start with the simple projects that work well. Oysters on straw, oysters on coffee. Get some success under your belt. Have a yield that you can brag about, take a photo of, and then branch out to the more experimental stuff. Always having fun, recognizing that we know so little, and that everybody can add to mycology, because it's one of the youngest natural sciences and one of the few things that we know. It's probably one of the biggest knowledge gaps in the sciences, especially in the natural sciences, if not the biggest knowledge gap. So, man, there's a lot we all need to do for the next few generations. So, anywhere you start, mushroom growing, yeah, have a good time doing it. Just start with the simple projects, work your way up. Don't feel like you need to master everything or have every tool. There's a lot of simple approaches. I always advocate for the appropriate technologies and the liquid culture stuff. If you don't know about that, look into it. Really easy, really fun, actually world-changing, in my opinion, game-changing. A lot of good stuff. Don't stick with the 30-year-old agar paradigms with the crazy lab and all the money you don't have, that kind of stuff. Things are changing. Internet's out there. It's a whole new world, and we live in an important and really fascinating time for human fungal relationships.
Yeah, I would say, and the big thing too is join local groups. I know like Seth Peterson's awesome. He's around here. We did grew some shiitakes, and they're doing microremediation stuff. I'm just blown away by how much mycology networks are out there. Like, Peter, where's a few good ones that you think are good ways to meet other people who are into mycology too?
Well, I mean, the easiest thing and the most widespread entry point is the just traditional Mycological Society, which is mostly focused on identification, toxicology, mycophagy, eating mushrooms. Some groups branch into cultivation and other topics. Of course, they bring in speakers that can speak on all range of topics. Those groups are generally more focused on traditional mycology, the harder science, the ID stuff. They certainly need everybody's support. They need the younger crowd. You'll go to these clubs, and often it's the older crowd, and that's not a bad thing. We need the younger folks to perpetuate this knowledge. It is incredibly important. I certainly support NAMA and all these societies. It's not where I spend my time because I'm doing something different, but I certainly value and honor all these elders that have added so much to our foundation. So certainly support these clubs, join them. That's where you're going to meet. It's the easiest entry point in most towns and places. Otherwise, historically, the mycophiles like us have been and still kind of are few and far between. In a big city like Oakland, you're going to get a denser concentration, so you can actually hang out with a few. Most small towns, you're like the only one, and if you're out there, you know what I'm talking about. But, we got the internet, we got ways to connect, not feel isolated. The radical mycology thing, we try to bridge these gaps, build a network, build a community amongst people that are interested in this, really compelling and bizarre and life-changing topic through a variety of means. We got a website that's, it's okay. We want to have better resources. There's always the dream of a forum that's not focused on psychedelics, which is what all the other ones are focused on. And our biggest thing is the Radical Mycology Convergence, which hops around the country every couple of years. And that's a big intersection point for people that are interested in all these other aspects of mycology beyond ID. We talk about ID, we talk about all the traditional stuff as well. We support and respect all that, but branching into the new eras that we are so rapidly, exponentially flying into, that's where I'm interested in, and that's what, obviously what I'm doing. But part of our work, and I talk about in the book, and it's been a thing, start a radical mycology group. That's what the Bay Area Applied Mycology group was founded by. It was my friend Maya who co-founded radical mycology with me. She started that group a bunch of years ago, and it's certainly an option. It's on the website. We actually say, hey, start a radical mycology group. If you don't have to call it that, whatever you want to call it, we'll put you on a map. We got like a list of people interested in this sort of approach to mycology, with the hopes that it's a way for people to travel and to meet and however, or say, look at our website and say, wait, there's somebody else in my town that's doing this. Otherwise, there's, of course, a lot of Facebook cultivation groups and ton of mycology groups on Facebook. And then the main forums is just if you want some sort of actual deeper community is Mycotopia and Shroomery. These are cultivation forums, mostly focused on psychedelics with sub-threads on edible, gourmet edibles and medicinals as well, and other tangential topics. But as with so many aspects of this whole field, there's not enough going on. Everybody can contribute. If you're a techie, if you want to add to the online face of this subculture, please.
Yeah, if only we had a fraction of the military budget to study the most important thing in the world. Wouldn't that be crazy? Oh, okay, as a quick plug for some of you guys who want to see more awesome, sustainable, regenerative design webinars like this, you can go on sustainabledesignmasterclass.com. Now we just put up all our other webinars on demand. We've got, John, just had Neal Spackman do a terraforming series. That was great. And we got all these other webinars coming up. We've got John Liu, that's going to be big. He's on the 22nd. Richard Perkins is next week. Peter Allen's on the 15th. January 5th, Kay Baxter. And we're going to keep going with this. We're going to get great folks. We'd love to have Peter back. And, of course, maybe one day we're going to have Paul Stamets, too, do a really big mycology one as well. But enough of this pitching stuff. Let's get back to more questions. Peter, if you got to go, let us know. But I like to stay on for people who still have questions.
Yeah, maybe like 10, 15 more minutes.
Yeah, sure. Why not? Okay, but yeah, I'll keep it recording. All right, so let's get back to questions, shall we? So Matt Mendez he says he's interested in using micromediation to help stop soil loss from erosion caused by development and road construction along a creek. Can you please speak to this?
We usually think about with erosion control, trying to get plant roots in there, to hold it together. But as I say, what's really doing the gluing? It's that glomalin, the Arbuscan mycorrhizae. Plant roots, tree roots hold big boulders and stabilize the big chunks. You got bacteria and fungi creating the micro and macro aggregates that really hold together soils and support the roots and all this stuff. So you got to bring in your plants. There's a lot of stuff written on erosion control with plants. What I add to that mix is don't forget the fungi. Right? You got to bring them in, and they're going to do that work as we've been saying.
Okay, so quick question from Alex. This one's interesting. It's all about enzymes and mycorrhizal and enzymes together. He says he feels as though enzymes are the key to ecology. They're the catalyst to decomposition and eventually the release of 10 times as much carbon dioxide than human activity. They also have the ability to break down anthropogenic xenobiotic toxins. Whoa. They are being used to create novel, patient-specific antibiotics by Trad Cotter. Where can I find the best information on enzomycology or enzyme mycology?
Fungi fundamental. It's kind of what I said at the beginning of the talk with how mycelium grows and how it lives is by constantly adapting to its environment and releasing customized suites of enzymes and acids and antibiotics and all kinds of things to defend itself, defend its food, and to digest its food. And whether that's food is a living insect, a dead piece of wood, or a xenobiotic, persistent, carcinogenic, toxic compound. Many fungi, giving the option of survive or die, will adapt and evolve rapidly through epigenetic analysis of its environment and respond by releasing the customized suites of enzymes. This is just like a fundamental concept of fungal growth in biology. It's how they live. It's how they eat. It's what they do all the time, all the day, every day. So, everything you would read about fungi will mention this. Maybe your question is, how do we get the fungus to customize its enzymes to what we want it to do? So if that's your question, then yeah, there's a lot of practices I detail in my book. We can introduce chemicals and microbes to the fungus. These are well-known concepts, and the fungus will respond to those microbes and the chemicals in tow. And as long as it's not stressed and it has the time and it's not being bombarded with a bunch of other stressors, it will learn very often to eat that thing. And then we can harvest those enzymes and utilize them in the raw form and concentrate them, or just bulk up the mycelium and it'll just kind of keep doing it as it grows. So these are practices I thoroughly detail in my book. It's a pretty fundamental concept. Testing those enzymes and knowing exactly what they are and their structure. That's enzymology is really elaborate science, and it's really hard to figure out the structure of an enzyme. But we can at least do these practices, and we know that that's essentially what it's doing, even if we don't know exactly what the compound is or what we could call it. Probably a lot of these compounds we don't even have names for yet.
Yeah, just this, most, 80% of what you're talking about was totally new to me, so my mind is continuously blown learning about this stuff. As a cool follow-up from our last webinar, Nicholas Netian was a farmer in Cyprus. He said that he tried all these different cultivation techniques for his olive trees, and he said the one that grew the best for his most healthy olive oil was the one that he inoculated with mycorrhizal fungi. It was the one that survived in some of the most extreme areas, and as a result, he has the world's healthiest olive oil. Have you heard of any other kind of extreme examples where fungi inoculated soils help people in the deserts?
Well, I love that story, and if there's any documentation of that, I'd love to see it and share it. At the same time, I'm not too surprised, right? This is so much of what they do, and I hate to beat a dead horse or whatever, but just hopefully we're all drilling this in and getting the point. It's a pretty big one. As far as others, kind of surprising, amazing stories like this, I don't know any good ones from the desert. Maybe they're out there. Again, I don't live in these parts of the world. I've never visited, that part of the world, so I'm not even really sure what common practices look like, or even, what that analysis is. I have plans to go to that part of the world and do some teaching in the not too distant future, but as of yet, I don't know.
Cool. All right, Tune as a question that he says, when's the next time you're going to Europe for talks, and talks or to give a workshop?
Well, the first time I'll go, I was hoping to say like go this year, then I was thinking next year. The way things are panning out, I definitely want to make a trip overseas. I got a lot of ideas and contacts, and a lot of potential good things could come of that. But I want to do it right. I want to make sure it's lined up and all these types of things. But so most likely looking at maybe 2018, probably a really extended trip and tour, and a lot of hopefully really good projects to come from all of that. So just stay tuned, to the social media. This year, I'm mostly focusing on a lot of projects, things I'll be announcing over the months, and other new things. And then hopefully next year, 2018, going international in a lot of ways.
All right, so it's been about 2 hours. 2 hours. We've had 160 people out of 250 people stick all the way to the end. You guys are awesome. Thank you.